All right. So we're going to do part 3. This is our genetics. I almost skipped this and decided to move forward into a different section, but I do think it's good we cover it. We talk about hemoglobin in here, phase 1, phase 2 detox, and purine pyrimidines. So I do think it's important that we look at it, but we might not spend quite as much time. I'd say this would be lower yield than the first 2 lectures. So nucleic acid metabolism, your purines, your pyrimidines, and more. First, a nucleoside is just your base here. Nucleotides are once we add phosphate. So nucleotide ties the base to a phosphate. Okay, so nucleotide phosphate group added to our nucleoside, which is our base. How many phosphates we have gives us this MP, DP, or TP. Okay, so our purines are aspartate and glutamine. These are our 2 purines, or I'm sorry, adenine and guanine are our purines. Adenine utilizes aspartate as one of the main amino acids that builds it up. Guanine uses glutamine. It's one of the amino acids that contributes carbons to it. We can see all of the carbons that are contributed to a purine base here. So this is a purine base. We see carbons being contributed from glycine, from formyl F4, from glutamine, a carbon dioxide, aspartate, another formyl F4, which is tetrahydrofolate, so a folate molecule essentially, and glutamine again. So it's kind of a combination of multiple sources that builds up our purine molecules. And then we have the 2 specialized versions here, adenine and glutamine. Okay. We look at purine synthesis. We are going to start, I need to move this, there we go. We're going to start here. All of these acronyms have definitions down below. Okay, so ribose 5-phosphate plus ATP gives us PRPP. Remember that molecule from before, the 5-phosphoribosyl 1-pyrophosphate? And that was an answer on your exam before. That plus glutamine, and then we go through all these changes, right? We add all these components, all these carbons to build up our purine. From there, oh, did I get something? Oh, here we go. From there, we're going to add in another ATP molecule and a glutamine molecule, or a GT molecule and a aspartate to either get our A or our G, right? Depending on which direction we're going. And then we're going to continue to add phosphate groups on board until we get to our triphosphate form. And then we're either going to go into RNA, or our deoxy form is going to go into DNA. So remember, they have to become deoxygenated to go into DNA versus the non-deoxygenated form goes into RNA. So our 2 purines, adenine, guanine, same backbone till we get to here. Then if we add an aspartate, we get adenine. If we add a glutamine, we get guanine. And then we continue to phosphorylate them until we get to either RNA, or GTP, guanine triphosphate, or adenine triphosphate. Or if we want to add them into DNA, we have to deoxygenate or deoxy, get them into the deoxy form, which we'll show how to do here shortly. Looking closer at this process here. So this is reduction of our ribose to deoxyribose. It does use 1 NADPH to reset. So we see our base form here of our purine. And then we have this hydroxyl group here. And then we don't have a hydroxyl group anymore. So this would be our ribose versus our deoxyribose. Deoxy meaning we took off that hydroxyl group. Uses ribonucleotide reductase and thioredoxin reductase. So 2 reductase enzymes to do this process. When we break down purines, so if we have purines and we want to break them down into other forms, why might we do this? Well, if we're in maybe we don't need energy, or if we've used a lot of energy and we have a lot of AMP around, a GMP around, and we're not doing a lot of DNA replication and we want to actually recycle some of these other carbons or components, we can see these being broken down. So our purine degradation process, so our guanine and our adenosine, they go on their own journeys initially. And then once we get to allopurinol, or no, once we get to xanthine oxidase on this side, sorry, we go from hypoxanthine to xanthine. Allopurinol actually can affect and block this process here, the drug. So we go from hypoxanthine to xanthine. That's our adzine process. This requires a cofactor, which is molybdenum. It's our first time seeing molybdenum. So xanthine oxidase, purine degradation is where molybdenum comes into play as a cofactor. The guanine side didn't need xanthine oxidase to make this guanine to xanthine conversion, but we do have an ammonium ion that is released, an ammonium molecule that's released, which is a waste product. Versus this side, we have hydrogen peroxide as a waste product. So we're going to start to see some buildup of either oxidative species or urea-based, nitrogen-based waste. And so we're going to have to figure out how to deal with that. We'll find our nitrogen-based waste. We'll utilize the urea cycle. And then with our reactive oxygen species, we're going to need to utilize our antioxidants and our ways that we can neutralize these species, which we'll look at both these later. Xanthine then is the common kind of component that then is going to undergo xanthine oxidase again to become uric acid. And what's our classic condition characterized by the elevation in uric acid? Gout. Yep, exactly. So our rate limiting, or really the only enzyme we care about in purine degradation, is xanthine oxidase. Uses molybdenum. Byproduct, hydrogen peroxide. No, I had my little waste projects there. Yep, it uses it twice. To go from only if it's coming from AMP, right? It only needs to utilize it once if coming from guanine. Because if we see here, if guanine just loses this ammonia group, which when it does, it already is this molecule. Versus hypoxanthine, we see it's close, but this is a double bond here. Well, I guess the oxygen comes on board here. But this double bond here is attacked by oxygen on this side. Versus this side, we lose this molecule here and then water binds. So we don't, this is a much lower energy reaction. We don't need an enzyme to actually drive this forward. Versus this side, we need an enzyme to drive this forward, the xanthine oxidase. And then we need one here again. That's why to treat gout, we can often, or a uric acid attack, we give allopurinol because it blocks these processes at xanthine oxidase. So it's downregulates this enzyme. So then we could get some to xanthine from this side, but we can't make this last conversion step. And here we get stuck at hypoxanthine. It allows our body to metabolize uric acid and eliminate it. It does not actually itself eliminate uric acid elevation. It just prevents us from making more. And so then over time, as we get out in urine, that number will go down. And so it will help with a gout flare. Our pyrimidines, so our pyrimidines look much different, right? They look smaller. So we have cytosine and thiamine, C and T, right? C goes with G, T goes with A. So these are coming into place in our DNA and RNA. And then in RNA, we have uracil, okay? So cytosine, thiamine, and uracil. The origin of the bases in the pyrimidine ring are aspartate and glutamine. Those are the two origin, and then one carbon dioxide molecule. So it's much simpler. It's a much smaller structure. So if we look at synthesis of our pyrimidine structure, we have glutamine plus a carbon dioxide plus 2 ATP. We then are going to use CPS2, which is carbamoyl phosphate synthase 2. We get carbamoyl phosphate. We add aspartate, we get orotate, and kind of see we go from here down and down. So our enzyme we care about in this process is CPS2. Oops. We'll look at it here in a moment. Once we get all the way down, we form uracil first. If we then add glutamine, that's what gets us cytosine, and these can be used in RNA. Cytosine then can go under its deoxy form to be used in DNA. And the cytosine form can lose an ammonia group, and then it can add some folate group to form our tyrosine. So if you're going to track the process, you create U first, then C, then T for pyrimidine synthesis. I talked about the only enzyme we care about for this process is CPS2. There is a version of CPS1, so carbamoyl phosphate synthetase. If this sounds familiar to you, CPS1 is used in the urea cycle. CPS2 is used in pyrimidine biosynthesis. They're separate. Why? How do we know one's for one, one's for the other? The pyrimidine biosynthesis happens in the cytosol. So that's where CPS2 is, versus our urea cycle happens, this portion of the urea cycle happens in the mitochondria. So that's how they're separated. But they are the same enzyme, just two different types. I just wanted to mention a moment for 5-FU just because it's funny. 5-fluorouracil is an analog of either uracil or thiamine, so it can kind of gaslight or hide as both. And you'll see it can 5-FU can become 5-deoxy-UMP, so uracil monophosphate, and then can kind of dump into this process here and actually prevent us from going from uracil monophosphate to uracil or from thiamine monophosphate, so it can block this process here. 5-FU is actually a drug. We can use it for cancer drug. So you can do this exogenously, and it's a treatment for cancer. It's on boards too. Essentially a uracil that's tagged with fluorine. Some pyrimidine breakdown or degradation, it does need NADPH to break down. So when we're breaking down these processes, we need NADPH, messed up those words there. We start with our cytosine. This can be converted to uracil, and then thiamine is here. From there, we use an NADPH, and then we break things down. We hydrate it. We add some water to break the cyclic bond. And then we use carbamoyl beta alanine or carbamoyl beta amino isobutyrate. This would be, I guess, the only enzyme I would know, but I honestly wouldn't memorize an enzyme for the process. And then from here, we have an ammonia and a carbon dioxide on each. And we eventually get to malonate and methylmalonate. Okay. Where does malonate and methylmalonate come into play? Do you remember where we see those later down the road? Yeah, absolutely. Great. Those are our waste products. All right. Now, DNA replication, translation, and transcription. Review them on your own. We don't have time to do it here. I don't have time to go through it. The only reason I even say it, they have not asked questions about it until last NPLEX. They asked questions on it. And so when you're reviewing it, I would focus on just mainly the major players, the major molecules, proteins, processes. When we go into path and phys, our last lecture, we will kind of tie in some of those pieces when we look at pathology. So you will get some there, but I encourage you to look at it now on your own because I don't want you to wait till week 4 to look it over. But I would review replication, transcription, translation, just so you have a good overview process of what this looks like. I think on the handouts, the student handouts, there might even be images there, like some basic images too of those processes. I forgot if I included those or not. Okay. So now we've looked at the pyrimidines and the purines and their breakdown. That was our DNA RNA components. I want to look at protein digestion. So we've looked at amino acids already, so the buildup of proteins. So now we're going to kind of break them down. We've broken down carbs, we're going to break down our proteins. So remember, when we consume food, let me move this back up. We consume food, we get into our stomach, we ate carbohydrates. In the stomach, nothing was happening. With proteins, it's different. Nothing happens in the mouth outside of mastication. Go through our esophagus, we get to our stomach, and then HCl is going to come into play. It's going to start to break down those proteins, the HCl, that really acidic space, as well as the churning and pepsin. We're going to release pepsin, which is going to kind of continue that acidic space in the stomach. From there, we're going to see we get to peptides. We go from proteins to peptides. So we've unfolded things, we've broken things down, we're in peptide form. Then the pancreas will secrete bicarb, trypsinogen, chymotrypsinogen, proelastase, and procarboxypeptidases, A and B. Why are they the ogen form or the pro form? These are both saying these are like zymogens. These are the inactive forms of the enzyme. they have to be converted to their active forms. And so we'll look at that process here. It's on the left. All of these pancreatic enzymes are going to break down our peptides to our di and tripeptides and even some amino acids, thanks to our aminopeptidases. And from there, our di and tripeptides can actually get taken up into the cell. So this is the one area where we can actually take up something that's not one single amino acid into the intestinal cell. And then from there, it can get broken down further into individual amino acids before it can go into the blood. So they can't leave the cell until they're individual amino acids, but they can get taken out of the lumen in a di and tripeptidase form or peptide form. Okay, great. So when we look at our proenzymes or our zymogens versus our active enzymes, we have pepsinogen, which gets converted into pepsin in presence of hydrogen ions, so in the acidic state. So when we secrete acid, pepsinogen becomes pepsin. Then we have trypsinogen when we have enteropeptidase, a hormone secreted that's going to convert into trypsin. Trypsin then converts the next three. Chymotrypsin, proelastase, and procarboxypeptidase all become their active form thanks to trypsin. So when in doubt, trypsin's your answer, right? Because it's utilized as the converter, the activator in most of these protease processes. That's our big picture of proteins. Let's look a little bit closer, shall we? All right. So, repeat that, please. Yes. Pepsinogen gets converted into pepsin by acidic states hydrogen ions. Trypsinogen gets converted to trypsin by enteropeptidase. And then trypsin converts the last three. Trypsin activates chymotrypsin, elastase, and carboxypeptidase. So when in doubt, trypsin's your game. Trypsin's what you choose for the activator of your proteases or your protein breaker downers. Of course. I talk fast, GG, so you guys slow me down. Get excited about proteins. All right. Enzyme and amino acid preference. This is likely too deep for NPLEX. So I probably would, I think I even deleted the slide in one form of this talk and then I added it back. I couldn't decide. So sometimes there's certain enzymes that will have a preference for specific amino acids where they'll target or cut the actual peptide itself. I would not waste your time memorizing it for this test. I just want you to know it clinically and just in your brain that like some amino acids have preferences for trypsin, some for chymotrypsin, some for pepsin. I have never seen a question that's that in-depth on NPLEX in regards to this polypeptide, the polypeptide chain, and asking you who would make the cut between aspartate and arginine, what specific protease. I don't believe that will happen. It's more likely they'd ask you the order of operations, who's being released first and what they're doing. But it's here if you want to get into the weeds. All right, so after the peptide is broken, we will see transamination occurring in the cytosol. So I mentioned peptide is like the small protein form, the unfolded protein form. Then we're breaking it into individual amino acids or di or tripeptides. Once we get down to these individual amino acids, we're going to transaminate. What does that mean? We're removing the amino group, right? That's all transamination means. And this is going to happen in the cytosol of the cells. So we can see here that glutamate is we're going to remove this amino group and we can become alpha ketoglutarate. This utilizes pyridoxal phosphate and pyridoxamine pyrophosphate. Pyridoxal phosphate is what vitamin? 6. Yeah, vitamin 6, B6. So transamination always requires vitamin B6. Okay. You also can go in the opposite direction, right? So we can see that oxaloacetate to get pyridoxal phosphate again, so we can continue to transaminate. Oxaloacetate, we could take that from the Krebs cycle using the malate shuttle, right? So oxaloacetate in the mitochondria can leave the mitochondria via the malate shuttle in exchange for malate. And then oxaloacetate in the cytosol. It can then get converted to aspartate instead of becoming malate again and going back. And if it does that, it's going to be able to hold an amino group. So aspartate and glutamate are really good amino or nitrogen harvesting amino acids. So they can take excess nitrogen groups and hold onto them if our ammonia levels or ammonium levels are getting too high, if the urea cycle is too taxed. And we'll see that come into play here with our liver in a moment. Yes. Glutamate and aspartate, if we're looking at these in the opposite direction. So we're talking about transamination, removing an amino acid right now, right? Or removing amino group right now. But if we go in the opposite direction here, from oxaloacetate to aspartate, aspartate and glutamate actually be good amino harvesters. They can hold on to excess amino groups. So if the body has an elevated amount of ammonia or ammonium and the urea cycle is too taxed, aspartate and glutamate can hold on to excess amino groups to help decrease that taxation. Yeah, no, that's a great piece. Great question. So what's the connection to liver enzymes? So transaminases, I mentioned they use vitamin B6, peroxidyl 5-phosphate or PLP. And so we have our alanine aminotransferase and our aspartate aminotransferase. Aspartate, yay, this one right here, aspartate aminotransferase. But both of these are utilizing, all these aminotransferases are using B6. But alanine aminotransferase can actually take pyruvate to alanine or back and forth. It also can take glutamate to alpha ketoglutarate. And aspartate aminotransferase can take aspartate to oxaloacetate, back and forth, or alpha ketoglutarate to glutamate, either way. And these are our liver enzymes, ALT, AST. So this is happening all in the liver. This is what we measure. We're looking for liver damage, liver stress, liver strain. They're normally inside the cells. So that's how we can tell if it's liver stress or liver damage, because if they're in the blood at high levels, we know that our little hepatocytes have released them into circulation, meaning those hepatocytes were damaged. Or else those wouldn't be in the blood. So we're not harvesting them from the liver. All right, so more on passing the amino hot potato. All amino acids will participate except for lysine and threonine. Transaminase enzymes participate probably the most. So we see aspartate, alanine, and gamma-glutamyl, so glutamine transferase. All of these are helping to pass the amino hot potato back and forth between all amino acids. As I mentioned, though, the two previous amino acids are the best at holding on to those extra amino groups if needed, when times of need. Lysine and threonine do not participate. They do not help with the amino hot potato. Lysine, because it already has an amino group that's charged. Threonine, I don't know, it's too small, I think, to house another amino group, potentially. When we go through oxidative deamination, so this was, we went through first, over here, transamination. Now we're talking about oxidative deamination. Okay, so different processes. This occurs in our mitochondria. Glutamate becomes alpha ketoglutarate. And it's going to release NH3 as ammonium, NH4. It uses glutamate dehydrogenase, which can use either NAD or NADP for a cofactor. When we do this process, it's in GDP or ADP or low energy states when we need energy, because then we're putting alpha ketoglutarate into our Krebs cycle. When this is downregulated, this is in high energy states, when we maybe are saying, okay, we have lots of energy, let's build some muscle. Let's get some amino acids here so we can build up our muscle in our body. So we don't need as much Krebs cycle. We have tons of stuff working for us. We can get more amino acids. We can go this direction instead. Oxidative deamination is going from glutamate to alpha ketoglutarate. We take off this amino group. We add this double bond, and this can go back into the Krebs. And then we get an ammonium ion as our byproduct. You'll see all these processes are going to start building up ammonium or ammonia. So it's leading us to our urea cycle. Again, looking at oxidative deamination, if we wanted to further kind of deaminate glutamate, or in the opposite direction, we could add another NH3 group and go from glutamate to glutamine. So in this picture here, we are talking about going from glutamate to alpha ketoglutarate by removing an amino group, but we can go the other direction, like I said. And so this is showing the other direction, from alpha ketoglutarate to glutamate to glutamine, taking our amino groups out of our muscle and putting them into glutamine, who is a holder, a storage molecule for us. Glutamine can go through the blood then and can go into our liver, where it can get taken into the liver and get broken down. And then those ammonium groups will go into the urea cycle and be excreted via urine. So again, alpha ketoglutarate takes the ammonia group, glutamine takes the ammonia group, glutamate, then glutamine. We're putting in an NADPH, we're putting in energy to go this direction. Glutamine can go through blood to get to the liver, where it then can become, it can go in the reverse direction in the liver, glutamate, alpha ketoglutarate, using the same enzyme, GDH. And then from here, our amino groups can go into the urea cycle and be excreted via the urine. Alpha ketoglutarate, so the alpha ketoglutarate that's in the liver can go back in because it's in the mitochondria, so it can go back into the TCA cycle. Yep, exactly. If we don't need it for, if we don't need to go like back in the reverse version for whatever reason, it'll just go back into the TCA cycle. It's already there. Yeah. So why do we ever break down proteins? We do when we need to use amino acids as fuel, specifically during fasting. So in a fasted state, muscle gets broken into protein, gets broken into amino acids, and then we have our TCA cycle. We're going to pump out glutamine because we've taken alpha ketoglutarate, we've converted it into glutamate and then glutamine. That then can go all the way through our kidney and our blood because we're filtering it through our blood, or it could go through our gut, and then we're going to convert it here. We convert it up into alanine, which then can go into the liver this direction. Or we can do muscle breakdown to alanine directly, can go into the liver, become urea, become glucose, become ketone bodies. Or we can break down our other amino acids and they can go into the liver, urea, glucose, ketone bodies. Ketone bodies, muscle kidney, glucose, brain red blood cells, urea, excreted. All right, how well do we eliminate extra urea and ammonia from the body? That we do it via the urea cycle. So the urea cycle is unique in that it occurs in the mitochondria and within the mitochondria and the cytosol. So this is another cycle I think we should know. If you're looking at the cycles, glycolysis, Krebs, oxidative phosphorylation, those are your most important. Urea cycle's up there. Urea cycle comes on the NPLEX a decent amount. So I would say like this would be another cycle to at least not ignore. So we start with the need. The purpose of our urea cycle is to try to get rid of these extra amino groups. So we have too much nitrogenous waste. So we take carbon dioxide and water and we create bicarb, and then we add bicarb with an ammonium group, an NH4+ group. We add those together and we're going to create carbamoyl phosphate using carbamoyl phosphate synthetase 1. Remember that CPS molecule I showed you earlier? Well, this is the friend inside the mitochondria that's involved in the urea cycle. The other one involved in our pyrimidine purine synthesis is in the cytosol, CPS2. This is the one in the urea cycle. To do just this tiny combination, we use 2 ADP. So this is a super energetically unfavorable reaction. Okay, we're using a lot of energy to get rid of nitrogenous waste. From here, our carbamoyl phosphate, we're going to convert it using ornithine to citrulline. We use ornithine transcarbamylase. This is not the rate limiting enzyme. This would be your CPS1 is your rate limiter. Citrulline, the reason we have to do that is because citrulline can go through this membrane and go into the cytosol where we complete the urea cycle until we get back to ornithine, which can go back into the mitochondria. So citrulline can become a aspartate, citrulline plus aspartate, our amino acid, can become arginosuccinate. We have to use another ATP, pulling off 2 phosphate ions. So again, super energetically wasteful. Arginosuccinate, we remove a fumarate and we get arginine. And then arginine, under using arginase, we get urea as the waste product, which can go into urine, and ornithine, which can go back into our mitochondria. And we have water here too. If you wanted to choose one other reaction that's important, I would say the arginine to ornithine, because that gets you urea as your product. So your first and your last reaction, those would be the 2 I would think about. Energetically unfavorable, we do it because we have to, right? The body doesn't like to do energetically unfavorable things unless it absolutely have to. We cannot have extra nitrogenous waste build up in the body. That's how we get things like, oh, what's it called? When our liver becomes toxic and overridden, and hepatic encephalopathy. I knew I'd get there. Liver, brain, inflammation. So you have a patient, they're an alcoholic, they have liver fibrosis cirrhosis, they've been drinking a lot more, even more than usual. They start noticing decreased urination or really dark urination. And then next thing you know, or they can't urinate, then they're in your ER and they're altered. And so first thing you're gonna do is check an ammonia level. Their ammonium level will be through the roof because they're not eliminating nitrogenous waste. So then if you need to, you cath them. And then you can give them medicine to try to kind of bind to and utilize and get rid of and eliminate the ammonia. And then after that level goes down, totally back to normal mentation. So the delirium or delusion completely goes away. But they can look like, they can mimic stroke, they can mimic dementia, they can mimic like almost like a schizoaffective presentation, depending on how the patient presents to the ER. Can be fast. It can be within like hours, days. That can happen, and it can lead to like that severe hepatic encephalopathy quick. Once you stop the elimination, that process, it'll escalate quickly. The process to get there, that would take more time. But sometimes a lot of those patients, they might not necessarily know that there's decreased urination until they're already altered. And so then they come to you altered, but they didn't even like recognize the signs, the warning signs, because they're typically used to being dehydrated and having less urination and having darker urination. There's other reasons that are not associated with alcoholism or fibrosis or cirrhosis of the liver that you can see a hepatic encephalopathy from this reason. That's just the most common like presentation, the easiest one to, I think, wrap your head around. No, we check a lot more. We'll check liver enzymes, we'll check electrolytes, we'll check blood sugar, and we'll do a head CT typically to make sure there isn't any like concomitant actual stroke, because any patient that presents that way will probably code stroke them first in the ER. But ammonia would definitely be one of the things we'd order in that first round of labs if we suspected. Yeah, good question. Well, fun ER lab tip for me to you. First patient like that, I was like, oh my gosh, it's a stroke. And I was like, one of my first days as a resident, I was like, it's my first stroke patient. Okay, I'm ready. And I was like completely clear. And I was like, well, I don't know. And they're like, will you check an ammonium? I'm like, no. What? They were completely altered, not responsive, like totally faced. They weren't like, they didn't have a droop. But they weren't like able to really respond to commands. And family member brought them in because they like came over to visit and found them that way. And so they were like, oh, they're having a stroke. And so that's why we stroke coded them. And then they weren't. And so then we got a little bit more history and was like, okay, we got alcohol level, and then we got an ammonia level. This is a good case. You could potentially have permanent liver damage or destruction, and similar to brain damage and destruction. So that buildup of those waste products can be pretty damaging over time. So you want to catch it quick. It's not going to cause like the same quick damage as like a bleed or something in the brain or ischemia. It's not going to lead to that level of necrosis, but you can see a progression of like necrotic tissue if it's left to its own devices for too long. Because ammonia ions, right, and amino groups, they all have a +1 charge. So we think like pH being affected. We think like so not just blood, but also like our cells, membrane issues, cellular permeability, neurotransmitter signaling, action potential. So you just kind of have this downstream effect that starts to happen. Yeah. Great. High BP as well. It depends. I mean, likely these patients would, because most of the time, well, at least the ones I've seen, they've been all ETH related, so or ATOH related. So yes, but not necessarily, because if it's been happening for an extended period of time, they could be low blood volume if they're dehydrated, or they may have other concomitant things going on that kind of has their blood volume or their blood pressure drop. So it really depends on the patient, but I think most of the cases we saw hypertension as well, but nothing too significant, nothing that exciting in the message of ER. Things that would be scary for us in primary care, but nothing that they cared about in the ER. Like, oh, 180 over 120, that's fine. Okay. You're dying. All right. This is just the words. This is exactly what I said. Your rate limiting step, like I mentioned, is CPS1. It can be stimulated by arginine. It can also be upregulated by acetyl-CoA and glutamate. If you're into like learning where the amino acids fit in to our non-essential amino acid production, so the essential amino acids we have to eat, right? We can't produce them. Our non-essential amino acids we can make. So this shows you the chart of kind of where these can fit in. If we're needing to make amino acids, we don't need to make as much amino acid as we do need to make energy. We do need to make amino acids to build up protein, to make all of the different proteins and enzymes in our body. So if we need to make those, these are the processes, the areas that they can go. So glucose to phosphoglycerate can create serine, glycine, and then cysteine eventually via methionine. Pyruvate can kind of move over to alanine, oxaloacetate to aspartate and asparagine, phenylalanine to tyrosine, alpha-ketoglutarate to glutamate to glutamine, and then that can also become proline and arginine. So you can see all kind of how they could fit in these different processes and procedures here. The breakdown, similarly, you can kind of follow them back into the Krebs cycle, right? So arginine, histidine, glutamine, proline can go to glutamate, and then alpha-ketoglutarate. Alanine can go into the blood, and it can come back into pyruvate. So threonine, tryptophan, glycine, alanine, serine, cysteine, all can become pyruvate. Aspartate and asparagine are the only two that can become oxaloacetate. Aspartate, tyrosine, phenylalanine can become fumarate. A couple interesting ones. Leucine is weird because it could become HMG-CoA, which we see HMG-CoA in our cholesterol synthesis. So that one's interesting. And then phenylalanine and tyrosine can become acetoacetate, our ketone bodies. I would not memorize these charts, although I do have to say NPLEX has tested you on the precursors of amino acids. So ones that might be helpful to relate is phenylalanine to tyrosine, methionine to cysteine, pyruvate to alanine, alpha-ketoglutarate to glutamate. These four, maybe these five. There. That feels better. Oxaloacetate, aspartate. So these five, I would maybe put in your brain as relatives together. Breakdown wise, I think the only cool one is this one, but I mean, they all could be pertinent, I guess. All right. Great. I want to touch on amino acid or hemoglobin and blood, and then we'll take a break. Okay, and we'll get to our last kind of chunk. The hemoglobin piece, I really want to just emphasize a few differences between hemoglobin and myoglobin, and just talk about a couple different types of the chains that you could get asked questions on. So myoglobin carries one oxygen molecule intracellularly. It has a single alpha chain and a high affinity for oxygen. High affinity means what? What's affinity? Kind of, yeah. Right, yeah, it's a tight, yeah, you kind of both say it together. So how how tight a bond can bind. So how strong a bond can be between one thing and another, between a molecule and its receptor. That's affinity. Versus avidity is how many times it can bind at one space. So like if I have high avidity, it'd be like if both my feet were part of like my binding mechanism to the receptor below. So I had 2 spots, or like now I have even more because I have 4 spots. That's high avidity. Versus affinity is like me like holding, like binding like this. That's less strong than if I bind like this, right? That's affinity. That's a tighter bond, okay? So that's your binding, your high affinity. So myoglobin binds really tightly, really closely to oxygen because it's only carrying 1 oxygen molecule. And we find myoglobin typically where in the body? Muscle. Yep, muscle, both skeletal and cardiac. Hemoglobin, for oxygen, carries it through the blood, has 4 chains. So 4 oxygen, 4 chains. 2 alpha, 2 beta chains. So that's your hemoglobin. So hemoglobin has quaternary structure. Myoglobin has tertiary structure. That's how you could think of those two structurally. Hemoglobin structure, there's 2 conformations. There's a taut and a relaxed. Taut conformation, oxygen's not going to be able to bind that well. Low oxygen binding ability. Partially because in the T state or the taut state, it's deoxygenated and the actual receptors are facing inward. So oxygen has a harder time getting in there to bind. In the oxygenated state, it's relaxed. So this is the R state, high binding affinity for oxygen. So if they mention the T state of hemoglobin or the R state of hemoglobin, they just mean taut or relaxed. Taut, not good oxygen binding. Relaxed, great oxygen binding. In the actual binding site of hemoglobin, there's a glutamic acid present. So a question that's come up on NPLEX before is what amino acid substitution takes place during sickle cell anemia? And the answer is valine replaces glutamic acid. So we have an acidic structure that's then replaced with a hydrophobic non-polar structure, valine. So when that happens, it's going to completely change the binding nature of that active site. So our sickle cell anemia active binding site is not going to be able to bind oxygen as well anymore because we've removed that active acid, that reactive molecule. We've replaced it with something that's non-reactive, non-polar. There are multiple types of hemoglobin. Hemoglobin A is the normal adult hemoglobin that we're talking about. Hemoglobin F is the only one I would really care too much about for NPLEX 1, and that's your fetal hemoglobin. It has 2 alpha chains like the adult, but instead of beta chains, it has gamma chains. That's commonly the question, is like, what's the difference between, or what chain makeup is fetal hemoglobin versus adult hemoglobin? And it has 2 alpha chains, 2 gamma chains. Adult has 2 alpha chains, 2 beta chains. The other ones are more associated with actually pathology. So hemoglobin A2, an increase in that is associated with beta thalassemia carriers. And then hemoglobin A1C is our measurement for diabetes. Iron binding. So we have 2 different forms of iron, ferrous and ferric. Ferrous is F2, ferric is F3. F2, ferrous, binds oxygen. It also can bind carbon monoxide. So you could have carboxyhemoglobin when a carbon monoxide is bound instead of an oxygen. That's not so good. Ferric binds CN minus, so methylmoglobin would be what that molecule would be called. It does not bind oxygen. So ferric iron does not bind oxygen. So in heme, we find ferrous iron. And this is the molecule of heme with its little pretty iron in the middle. When we talk about iron, the most of the time we're talking about it in relationship to oxygenation, tissue perfusion, those type of pieces. So this is a graph showing myoglobin and hemoglobin. When we have a left shift in the graph, so if these curves move to the left, that's what they're talking about, left shift in the graph, we're going to see an increased affinity for oxygen. So we're going to see that more oxygen is going to be able to bind to these molecules. So we're going to reach maximum oxygen saturation quicker. That's what this graph is showing. Okay. Partial pressure of oxygen is the measurement on the bottom. And then saturation with oxygen of these receptors is on the y-axis. So myoglobin is already pretty has a high affinity. It almost is directly upward. Hemoglobin has a little bit of a lower slope, but you can still have a left shift. And you can see that in tissues, we have a higher binding affinity for oxygen versus in the lungs, because the saturation is so high. So when we have increased affinity, hydrogen is reduced, we have high pH, carbon dioxide are reduced, 2, 3 BPG is reduced. I think I have a picture of what that is coming up. If not, I wouldn't worry about it. In a right shift, we have a decreased affinity for oxygen. All are in their taut form, when they're non-relaxed form. So we'll have increased hydrogen ion. We have low pH. This is referred to as the Bohr effect versus hydrogen ions being bound up. Elevated CO2 decreases or increases hydrogen ions directly, makes it more kind of acidic. And we have elevated 2,3 BPG. I think this binds between our beta units. I think it can bind instead of and block our beta units, but don't quote me there. So what happens in the lungs? How does oxygen leave hemoglobin? This is the Haldane effect. In the lungs, the oxygenation of hemoglobin promotes dissociation of hydrogen ions from hemoglobin. This shifts the equilibrium towards more CO2 formation. Therefore, CO2 is released from red blood cells, and then oxygen is taken back up. Because otherwise you'd say, like, why is CO2 released from the blood cells? Why isn't it just stay bound forever? Because we get to the lungs, and the lungs have all this oxygen present, and then red blood cells are like, oh my gosh, I want this oxygenation. The pH shifts, they get rid of their carbon dioxide and are able to bind oxygen. That occurs at our alveoli. And they just move downstream. They diffuse across the alveolar membrane down their concentration gradient. So you don't need any energy to do that at all. Only reason this is in my head is because I teach respiratory this week and structure and function. All of the nitrogen in heme comes from glycine. All the carbon comes from glycine and succinate in heme synthesis. I've heard this glycine one before, so when in doubt, I'd maybe associate glycine with heme. That one I've seen tested. If we cared, I don't think we should, but if we did, this was our heme synthesis pathway. The only reason is just to show it's glycine plus succinyl-CoA. And then our ALA becomes our porphobilinogen, which is one pyrrole, one of these little kind of triangular house-like molecules that are then going to eventually come together to form our hemoglobin structure, as you can see here, or a heme structure. This is the whole meal deal. If you're really interested, the only reason I find this chart helpful is for porphyrias. I do not believe any porphyrias are on your NPLEX I exam. So I think you can skip this slide for now for NPLEX I. If a porphyria is on your NPLEX II exam, then you might care about this slide. But for now, for NPLEX I don't think there's any porphyrias on your pathology. When we break down heme, so what pathology would be indicated by an increase in total bilirubin? Typically liver pathology. Direct bilirubin is bile excretion issues. Indirect bilirubin is Gilbert syndrome. Why do we care? Because heme breaks down into biliveridin, and then biliveridin breaks down into bilirubin. So that's why I'm talking about this at all. Heme breaks down via heme oxygenase and then biliveridin reductase. Heme oxygenase is kind of this first step. That's where oxygen comes in, CO2 comes out, and we get the iron kicked off. Once the iron's kicked off, that's when we can cleave the bridge and open it up. Again, these are lower yield pathways. I think the interesting connection is with the pathology. Heme breakdown again. So total serum bilirubin, this is just talking about the direct versus indirect forms. So direct bilirubin is the conjugated form. Indirect bilirubin is the unconjugated form. You have to calculate this number. If we look from, okay, red blood cells, hemoglobin, what happens? After 120 days, our red blood cells break down and then heme is exposed. We then will see that heme could be taken up again by different things like myoglobin or cytochromes can also give us some heme. From there, that will get broken into bilirubin. Iron and carbon monoxide is released. Then from here, we have bilirubin, which will combine with albumin. They'll move through, and then we get our bilirubin dichloronucleonide. Then it becomes bile, urobilinogen, and then feces. That's kind of like our excretion process. I think more importantly is just this piece here. My brain didn't keep in my mind that heme becomes bilirubin. I think that's the key piece. All right, I think we can do it. We're gonna try to push through for 4 minutes, okay? And then we'll break. Phase 1 and phase 2 detox. We're in the liver, we're working hard. Phase 1 detox, I want you to think of hydroxylate. Phase 2 conjugates. So if we have a xenobiotic or a waste metabolite in your diet or in peripheral circulation, we need to detox it. We need to stabilize it. We need to get rid of it. We can get it to if we can get it to the liver, we can go through phase 1 reactions, which is hydroxylating. So this could be a reduction, an oxidation, a hydroxylation, and hydrolysis. After phase 1, after we hydroxylate it, we have a primary metabolite. Then we go through our phase 2 reaction. This is conjugating. This could be adding a sulfate group, adding a methyl group, putting glucuronidation, putting some sugar on it. And then from there, that gives us our secondary metabolite. And that metabolite is what we can finally excrete. But phase 1, phase 2 detox is really just our goal of getting something that's a waste product into a form that can excrete it. But we have to go through 2 phases typically to get to a place where we're able to safely eliminate. So what can that look like? Here's some examples of enzymes used in reactions in phase 1 and phase 2 detox. So these are some enzymes used in the biotransformation of xenobiotic compounds. You'll see that there's quite like a variety, and also there's some that are just generic, like dehydrogenase, reductase. Like these are obviously generic and they're going to depend on the metabolite or the xenobiotic that's needing to be transformed. Okay. So we can see if we have the negative metabolite, we can see an oxidation or hydrolysis reduction. This is our phase 1. This is typically our lipophilic phase, our fat loving phase in phase 1. We then have our electrophiles up top, our nucleophiles down below, depending on what we did to it. Did we add a double bond with an oxygen? Did we add a hydroxyl group? Did we sulfonate it? Did we add an SH group? So what did we put on there when we added something initially? From there, moving into phase 2, that's when we're going to do our conjugation. So we're going to neutralize this component that we already added something to. So we add, this is a glutathione conjugation. So using our glutathione pathway, we added a sulfur group, we sulfonation or acetylations, we added an acetyl group, we added a glucose molecule. This is our hydrophilic phase, our water-loving phase. We have to go through a hydrophilic phase because we can't excrete something if it's water-fearing, right? We can't excrete something if it's lipophilic, because then it's going to have to be transported via some transport mechanism in fat. So we need to get it to be water-loving so that we can actually eliminate it via feces or urine, but typically feces. If you look at here, we have our cytochrome 450, our actual enzymes that you can see. And then the enzyme itself, our P450 reductase. So this is where we're living in our liver. This is you can see our little lipid bilayer here, super cute. We are going to use NADPH in this process. We are also going to use FAD. So we're using riboflavin. And we are oftentimes going to use heme as a stabilizer, potentially, in this process as well. This is our phase 1. Of these are all your CYPs that are significant. So your significant cytochrome P450 enzymes, they're inhibitors, inducers, substrates. Do not memorize this. In fact, like this would be good to say for later clinically, but on an NPLEX, just know P450 is the common enzyme involved. Most common. There's others. P450, cytochrome P450 is your most common involved. I would not do any more than that. Okay. That was genetics, metabolites, and some other stuff. Last piece, we're going to take a break for 10. We have kind of 2 more pieces. One that I absolutely want to get through, which is our clinical connections. This is where we're going to get through all of our fatty metabolism, our cholesterol, our cortisol, our steroid hormones, all those biosynthesis. So that's our next bet. And then at the end, we'll touch on vitamins. That'll be our last hour. Okay. All right. Let's take a 10. We'll come back at 11 after, and then we will continue to power